students. The cell-mediated immunity component of the adaptive immunity is taken care of by T lymphocytes. These immune cells in turn originate in bone marrow, then travel to thymus via blood and undergo antigen-independent maturation there. They possess a T cell receptor called TCR and an assisting molecule, namely CD3. TCRs, unlike BCRs or antibodies, cannot bind to the intact antigen. Rather, they respond to the processed antigen which is presented on the MHC of the antigen-presenting cell or the altered self cell. Kepler and Marek were first to demonstrate that a single TCR couples simultaneously with both processed antigenic peptide and the MHC molecule on which it is placed. This complex is known as the ternary complex. The TCR belongs to the immunoglobulin superfamily and is a membrane-embedded heterodimer with each chain composed of a proximal constant domain and a distal variable one as shown in the picture. The routine TCR has alpha and beta chains, although there is also found a gamma delta TCR2 among neonates or in unusual emergency conditions such as infections, etc. among adults also. Talking of the routine alpha beta TCR, each variable region contains 3 hypervariable regions or CDRs, just like it happens in immunoglobulins. Besides the extracellular region, both chains in the TCR molecule also contain a transmembrane region that is rich in hydrophobic amino acids, immediately followed by a short cytoplasmic tail region towards its C-terminal. TCR also exhibits vast variety just like BCR, following similar recombination mechanisms such as allelic exclusion, junctional flexibility, P-region nucleotide addition and N-region nucleotide addition, so on and so forth. Besides the main T cell receptor, there is present a coreceptor also on the T cell, which is CD4 in T helper cells and CD8 in T cytotoxic cells. In the ternary complex, these bind from the side onto the lateral portion of its pairing MHC2 and MHC1 respectively. The TCR is surrounded in space by a complex assisting molecule called CD3, which in itself is arranged as 3 transmembrane dimers, zeta-zeta, gamma-epsilon and epsilon-delta chains encircling the main structure, the TCR. CD3 provides help to the TCR for emerging on the T cell surface. The TCR also functions with the help of this CD3 that is bestowed with a substantial to long intracellular portion loaded with very characteristic motifs called ITAMs or immunoreceptor tyrosine based activation motifs, since they are rich in tyrosine amino acids. A total of 10 such ITAMs are found in a single CD3. Please refer to this picture. Its role is to carry out signal transduction as soon as TCR forms a ternary complex with the processed antigen placed onto the MHC of the presenting cell. Besides responding to the processed foreign antigen dished out on self-MHCs, T cells are also known to respond to foreign MHCs alone too, which actually forms the basis of transplant rejection. As a matter of fact, MHCs were first identified during MHC incompatibility induced transplant rejections by the recipients, which is exactly why These membrane proteins were named major histocompatibility complexes or MHCs. Guys, we, Brahmjeet and Archana, are thankful to all of you for watching. In our next video, we shall discuss how apoptosis or programmed cell death shapes up the final T cell repertoire. Till we meet soon enough, stay blessed and studying. Bye till next time.